Welcome to the Vitamin Professor podcast, brought to you by VRM Media, publisher of vitamin retailer and nutrition industry executive magazines, in partnership with Nutraland USA. I'm your host, Dr. Gene Bruno, Professor Emeritus of Nutraceutical Science and Chief Scientific Officer of Nutraland USA. Now, the purpose of this podcast is to help educate the dietary supplement industry about the science of nutraceuticals and other relevant topics. If you like what you hear and see, don't forget to like and subscribe. Now, let me tell you a little bit about our guest today. With over 25 years in the natural products industry and a Master of Science in medicinal chemistry, Sebastian Balcombe approaches natural ingredients and delivery systems discovery and development through the lens of an experienced entrepreneur and scientist. From in silico computational chemistry for compound discovery to clinical research and IP development, he has developed over 30 nutraceutical ingredients, including multiple liposomal and beadlet delivery systems, as well as the IP surrounding them. With his in-depth understanding of liposomal technology, Sebastian's most recent research has focused on how this novel delivery system works with natural ingredients, including the ability to increase bioavailability and stability of critical nutrients in the body. Sebastian's authentic passion, craftsmanship, and commitment to creating a superior customer-based experience have built his reputation as a pragmatic perfectionist in the industry. He is dedicated to helping people achieve a better quality of life and is committed to establishing new standards in product development, innovation, research, and quality control. Today, Sebastian is going to talk to us about liposomes and specifically Lipo Advantage. Welcome, Sebastian.
Thanks for having me. Nice to see you, Gene.
Nice to see you too. And for those of you who don't know, I have known Sebastian for a number of years, and I got to tell you, I think he's one of the smarter guys in the industry. So I think you're in for something special today. So, there's a lot to talk about, so I'm just going to jump into it. Let's start with this question. For our listening audience who may not know, what are liposomes and why are they important?
Sure. Always a good place to start, just get some of the general understanding. Think of liposomes as a sphere or a round shaped structure, and they'll be made up of one or more lipid bilayers. Typically, the lipids are phospholipids, like what you would find, when you think of something like phosphatidylcholine or phosphatidylserine. Those are phospholipids, and they're making up this one or more lipid bilayer. So think of it as a surrounding, a lipid surrounding of an ingredient, which is going to have a very large impact at varying degrees on the bioavailability of different ingredients. So that's probably the best way to visualize it.
Very good. So, with regard to liposomes, why is validation so important, and are there industry standards for liposomes?
This is something you have heard me talk about for years and years. We definitely have been so focused and passionate in bringing the attention to liposomes in the last 4, 5, 6 years, and really approaching it from the testing validation perspective. So much of the industry just jumped into talking about what are liposomes and how can they potentially improve the bioavailability of ingredients. But they completely skipped the most important thing, which is how do you even know that your ingredients are liposomal to begin with?
Right.
You simply cannot just say because you mix some sunflower oil with an ingredient that now magically you have liposomes, which is really what the majority of the industry was doing.
I've seen it time and again, Sebastian. I've seen products where they basically they take some lecithin or something and they blend it together and wave a magic wand and call it a liposome. And sometimes it's painfully obvious just in the labeling of it when you see a product that will say something like magnesium threonate, that is liposomal magnesium threonate, and yet it has exactly the same elemental potency of magnesium as liposomal. It's like that's not possible if it's a true liposome. So I mean, lying about this out there is unfortunately too common.
And it's where we address, it's where we spend a huge amount of time, because again, the industry, when I first started doing interviews and being asked about these questions, the first question was always, liposomes have been around for a long time. What makes Specnova's liposomes different? And I would have to just stop the whole conversation there and say, yes, liposomes in pharmaceuticals have been around since the 1960s, but they have not been around for a long time. Real true liposomes in our industry have not been around for very long at all. And how do I know that? Because we've been testing almost all of the industries now for the last 6 or 7 years to see what's what. And there are so few that have actually tested out to have liposomes present. And then even then, they're very low quality, they're very few, and they're very simple liposomes. They're nothing like the multi-layer liposomes that we do, which will have multi-lipid bilayers, as we show and prove and are fully transparent when we share our cryo-TEM images. So we really had to back the conversation up and say, companies need to validate and prove first that they're making liposomes, and then forget about getting into like the intricacies and the differentiation between liposomes. Just even prove that you have liposomes present
in the first instance.
Exactly, because then we went way further with it. Okay, now we started to really dial in our formulation. Okay, we need to improve the stability of the liposome. We need to find the optimal zeta potential, which is largely the attraction between the compound or the ingredient inside the liposome and the liposome itself. We need to increase, which I think is probably one of the most, if not the most important aspect of a liposome, is what is the percent encapsulation efficiency, which means how much of the ingredient is actually inside of the liposome and how much of it is freely outside of the liposome, because it's not going to benefit if it's just free and not inside
it's
the
going to be
liposome.
it's just going to be like it did
Exactly.
nothing to it, right?
And nobody was doing that at the time, where we set a standard at no less than 80% encapsulation efficiency, with many of our getting into 90, 92% encapsulation efficiencies, just depending on the ingredient, but a no less than 80% encapsulation efficiency, which is extremely high, and why we perform so much better when we actually do compare our liposomes to the very few legitimate liposomes
That
in
are
the
on the
industry.
market. So then how do you really go about validating the liposome? Is it just, you gave sort of that specification of what you're looking for. How do you go about actually validating?
We've built basically a gamut of 6 or 7 tests that we will do to full, and we're always adding and improving when we can, but we're going to do 6 or 7 tests to really fully not just validate the liposome, but characterize it fully as well. So the first thing that you always hear, and it is the first thing that everybody should start with, because it's not even worth doing those other tests if you haven't even verified that there's liposomes present. So the first test is the electron microscope, or cryo-TEM, TEM. This is the tool that looks inside of the liposome or not and tells you, are these lipid bilayers present or are they not? Or is it just a micelle? Or is it just nothing?
Right,
Which is
and it's
mostly,
extremely revealing. I've seen
yeah,
those photographs of those electron
yeah.
microscope scans, and it's pretty darn evident when
It
you
is.
look on
It's
whether it's
really cut and dry.
yeah, yeah,
It's, we always talk about, on the analytical chemistry side, there it's not so cut and dry when you're testing ingredients because maybe the method was wrong, maybe the prep phase was wrong, and it didn't even go into the analysis to begin with in the correct manner. It's much more straightforward with cryotem. There's a very specific way that a sample is prepped into this frozen, semi-frozen state into the cryotem. Magnification varies. Like we'll take images at maybe not maybe 7,000 or 20,000 magnification. So it's really cut and dry. They're either the liposomes are either there or they're not. And it's the first step that you need to do before you do anything else. And we do this every single week. We run anywhere from 5 to 10 samples. I mean, every week. So we're running 15, 20 samples a month. We've been doing this for 6, 7 years. Like, we have such a library of cryotem images of not just our different formulations, but what we see out in the industry. And another thing, before I even jump onto some of the other tests, to really say how we fully, characterize our liposomes, we've been pulling off our customers who use our liposomes. We've been pulling their products off the shelf and seeing are the liposomes still present in those capsules or in those
nice.
gummies. Was super interested because you really don't know, right? Once we've sent it to their co-packer, we don't know fully the conditions that they've gone through, and we've been so happy, and we've been just sharing this with our customers, where we tested our Mega Foods launched our liposomal glutathione, beautiful liposomes present. A bunch of Dr. Mercola products we tested, again, liposomes present. We did a physician's brand, physician design. They do a gummy. We actually never tested to see our liposomes present in a finished gummy, beautiful liposomes in the gummy. So now we're just pulling off store shelves and testing our customers' finished products as well. So it's been really, really fun and interesting for us. But getting back to your original question,
That's pretty
cryotem,
cool.
start with cryotem. You must test for encapsulation efficiency to know how much, again, is inside the liposome or not. And these are really the tests that make up our true liposome validation process. DLS is another thing that people may or may not have heard of, but this tells you, this is identifying the average size of the liposome in the formulation. And we're really trying to target 100 to kind of a 200 nanometer range because we
Okay.
know nanometer sized liposomes, they have better absorption at the small intestines level, they go into liquid better. So we really target for this nanometer range, in that kind of 100 to 200 range, and usually they're maybe average 150, 160 nanometers. So DLS is the tool we use there. We also do another electron microscope imagery, which is called SEM, or scanning electron microscopy, and that will visualize the outside of the liposome. It's not going to show you if it was a liposome and the layers in the inside, but it will show you, now that we know there's liposomes present with cryo-TEM, we're going to then photograph it from the outside so we can see how is the shape, is it leaking, is it spherical, like, do we need to improve on that? Because
Sure.
in a lot of ways they're like little balloons, and if they're odd shaped they burst and then the ingredient that's in there comes out. So SEM is another key test we do. This is something that we do that I don't know, well, most of those nobody does, very few companies do, I should correct myself to begin with, but we also do powder X-ray diffraction. This is where we're looking to see The if the formulation, if there's crystallization that's formed, you don't want crystallization formed with your natural product ingredients, especially things like CoQ10. So we're trying to reduce this crystallization and keep the ingredient again in the liposome. So we'll use powder X-ray diffraction. We'll also do just a simple optical microscope where we're looking to see, how did the formulation disperse into a liquid? Can we improve it, in its dispersion characteristics in liquid? Because think about these are all very practical tests. Like that is your stomach, which is liquid. How is it dispersing in your stomach before it gets into the small intestines, which is a major factor in bioavailability, along with, of course, permeation. So,
as I listen to you describe some of these tests, it makes it really evident why so many companies that offer liposomes, or say they do, aren't really validating them.
No.
To do it right requires a number of steps and would also reveal whether it's real or not.
Exactly. I mean, it's again, for us, it's 6 or 7 at a minimum tests for us to really characterize and validate our liposomes. It's not, you don't just stop at cryotem. But again, there's so few companies, like they might show some generic cryotem image on their website. How do you know what ingredient that was done, what lot number that was done on it? We're like doing this over and over. We're doing it on specific lot numbers, and we share this, with our customers. So there's no question that they're getting validated highest level liposomes in the industry.
Let me ask you, I think in our industry, people are familiar at least with the concept of liposomes, and they know that in some way it may improve bioavailability. In your particular liposomal delivery system, what have you found to be, let's say, sort of the improvement in bioavailability that you might see in a given nutraceutical? I mean, I know it differs from nutraceutical to nutraceutical, but you have kind of a range where you could say, use of this can actually improve bioavailability by X%?
Yeah, it's a really good question, and it's also something that we've really worked to raise the whole industry on this. So first and foremost, before I even address the bioavailability question, I want to make it clear that liposomes not only improve bioavailability largely through improving dissolution and permeation at the small intestines level, permeation, they also improve stability as they go through the GI tract.
Yeah.
And people don't think about that. They're like, when we liposomed our vitamin C, Vitasomal C, amazing clinical study behind it. We already knew from some of the in vitro work that we did that the liposomes had a dramatic effect on reducing the degradation of ascorbic acid in the small intestines.
Wow, and let me tell you something from, I won't say the company, but a company that I worked for some years had a vitamin C product, and dang, man, that you could see the degradation, the oxidation that would take place with that product before it even reached the body. It just on the shelf a while starts out kind of white, and then starts becoming a beige, and then starts becoming tan, and you're like, oh my god.
Yeah, exactly. People don't think about that with liposomes. Forget about even the amazing bioavailability. Take into account that if the ingredient breaks down and loses a very large percentage of itself before it even can be absorbed at the small intestines, then that's the issue you need to address.
Yeah, for sure.
So we, with vitamin C, there was a huge decrease in degradation of the small intestines. It was
nice.
I think it was close to 40%. I don't have the exact number in front of me.
Wow, that much, huh?
You lose a huge chunk of it in the alkalinity of the small intestines. So take that into account first and foremost. Now, to answer your question, I particularly was super frustrated with our industry on these bioavailability PK studies. I used to always talk about it with friends and just kind of have this internal argument in my head, which was, When you share blood plasma data, like your area under curve, Cmax, Tmax, that kind of thing, that is obviously important information to know, but it doesn't necessarily translate to a greater effect of that ingredient. So we wanted to translate bioavailability and whenever possible in our PK studies to efficacy and an endpoint
Wow.
efficacy,
Yeah.
outcome. So and then the other thing before I even go on further was I found that most of the PK studies done in our industry on liposomes are very like disingenuous for a variety of reasons. One is when you look at the timestamp that they do blood draws, they're usually from 0 to maybe 4 hours to 6 hours. That's a very small window. What happens over 24 hours?
For
Of
sure.
course, you're going to get a big spike in those first few hours. But what if the drop off, and this happens in many, many ingredients, is so great that there's no net increase in absorption of that ingredient over 24 hours. So right from the start, I was like, all of our PK studies, they're going to be blood draws over 24 hours. They're not going to be these tiny little windows. And then number two was some of these PK studies, they use insanely high doses of an ingredient that we don't use. And why
Right.
do they do it? Because you can see it in the blood easier in these high amounts. But people typically are not taking 4, 6, or 10 grams of ascorbic acid, vitamin C at one time. So first and foremost, we wanted to do PK studies that were 24 hours, which we did. And then the other thing was use a dose that people use. So in this case, we use 500 milligrams of vitamin C. People typically take 500 to a gram. And then try to tie it to an efficacy outcome, the big
Yeah.
change in blood plasma. So we actually looked at concentrations of ascorbic acid in immune cells and white blood cells and leukocytes to show, look, we did this big increase in blood plasma levels of vitamin C over 24 hours. It was higher and more sustained than non-liposomal C. But then we also saw a 20% increase of that vitamin C getting in from the blood into the immune white blood cells. And like,
That is huge because I, I can't tell you how. Well, you probably know this as well or better than I do. That so many times in any bioavailability type study or PK type study, that's really just what they're looking at. What's the improvement in bioavailability, and not asking the question, okay, you have improved bioavailability. Does it do anything once it gets there?
exactly.
And that's all too often. I see it throughout our industry, and I remember for years it seemed like, creatine monohydrate, for example, lots of research on it, and then everybody would come out with a new sexy form of creatine, and the big thing was, oh, it's better absorbed, greater bioavailability, and no one asked the question, okay, so does it do anything once it get? Do you have any proof that it'll do anything? And
Yeah.
to hear you say that you try to do that translation of bioavailability to efficacy, that is huge.
Yeah.
I can't recall. It may be that there are others out there that are doing that, but I really can't recall off the top of my head any others that are doing that. So, kudos to you guys for taking that extra step. That's awesome, man.
Yeah, thank you. I mean, I like a lot of these things from the ingredients we do. I'm just obsessed with making them perform better for the end consumer, but I'm also obsessed with making our studies better and more relevant to the end consumer as well. Because you remember, Gene, I mean, it wasn't that many years ago when we were seeing, we'd go to these trade shows and you would see curcumin 5,000, 10,000 times in
Yeah.
I only need to take a milligram now. Like it's
Yeah,
just
right.
it's so absurd. So translate it to an endpoint so you know really how that effect is happening. And your creatine example is actually perfect. and it's the other end of the spectrum. So early days when different forms of creatine were being compared, for example, like creatine monohydrate versus citrate versus another pyruvate, you
Yeah.
could see again in these early hours of this PK study that these other forms got into the blood quicker, so they were elevated higher and faster than monohydrate. So people were like, okay, that means they're superior. No, then you read the whole study and you look at muscle creatine retention concentrations,
Kind
and
of an important.
they're inferior because there was a bottleneck. You could only get so much creatine from the blood into the muscle cell with a transporter. So you actually didn't want to be flooding creatine into the blood. You wanted to slow it down, but you can easily misinterpret and market these bogus results and say this is better because it got into the blood faster and higher levels. And you completely left out the fact that muscle creatine concentrations were actually lower in those faster absorbing creatines than creatine monohydrate. So
Yes.
that's a
Yes. Yes.
good example.
Excellent. So
Yeah.
back to that original question then. So what are you seeing as the range, the bioavailability range of improvement?
Yeah,
With
so
real liposomes.
like the vitamin C study, over 20% improvement in concentrations of vitamin C in white blood cells, in
Nice.
leukocytes,
Where it counts.
where it counts exactly. So um, it will vary from ingredient to ingredient, but they're all kind of in this range, this 20-30% range. CoQ10 had our published study on liposomal CoQ10, one of, if not our strongest studies. People know CoQ10 has bioavailability issues. They also
Yep.
know it forms these nanocrystals. It crystallizes with itself, and then when it's these big blobs, it doesn't absorb well in the small intestines. The liposomes help separate that crystallization from happening. Of course, berberine, our study's not published on berberine, but again, in that 20-30%. And people, why I mention this, people need to take and understand this. You don't simply compensate by taking 20 or 30 or 40% more of that ingredient. We're not talking about getting it into the blood. We're talking about getting it into a tissue, having an effect. It's a much more important
More of
metric.
an absolute bioavailability kind of issue.
Exactly. And then with a lot of these ingredients, taking more and more, there's limitations at the intestinal level as well. There's transporters. There's only a certain rate that, like it's not just taking more to offset that percentage. It doesn't work that way.
Right.
Yeah.
Cool. Well, that's very, very interesting. And so can I've seen in the industry all kinds of ingredients that are marketed as being liposomal, and so here's the question: can just any old nutraceutical be made into a liposome, or are there restrictions on that?
Most vitamins, minerals, small like short chain di or tripeptides, most plant compounds can be liposomed, and they do benefit to varying degrees. One of the biggest challenges between them will be what is the load of that active that you can get into the liposome. I'll use two extreme examples. Vitamin C, we can get an extremely high load, 70,
Right.
75%. A massive molecular weight structure like vitamin B12, methylcobalamin, we're in the 3 to 4% range because it's so massive, it's taking up so much space within the liposome. So it does vary, but for the most part, most small molecules are perfect to be liposomed. Doing large peptides, that's different. It depends. But for the most part, small molecules across the board with different load percentages are like the dream, thing for liposomes to load into a liposome.
And does it make a difference whether that nutraceutical is a water or fat soluble molecule?
It's part of it. It's definitely part of it. The polarity plays a role, molecular weight plays a role. So all of these things for sure, play a role in terms of like the load percentage that we can get in there. Yeah, absolutely.
Okay, cool. So let's talk a couple of specifics. I mean, you've done some studies. I'd like to hear a little bit. Can you tell me just a little bit about your berberine liposome study? What you found?
Yeah, the berberine study, I can't talk about too much because it is in peer review
Got it.
right now. But it was actually our strongest study out of all of our liposomal PK
Really?
type studies. That and then the CoQ10, and then the vitamin C studies, incredible gold standard. But like the biggest increase that we saw were those two, the berberine and the CoQ10. Just high level without getting into too much details. Just think about berberine. Typically, people that are seriously taking it for blood glucose control, they're taking 500 milligrams usually 2 or 3 times a day. So they're taking a gram. Yeah,
1,500 milligrams a day here.
exactly. So that's a good size dose, and a lot of people can have GI distress from it. It's obviously there's always compliance issues when you're having to take something 2 or 3 times a day. So liposoming it is a beautiful way to reduce that dose, get the customer compliance better, and then most importantly, you're making it more effective. You're getting a higher percentage into the body. So berberine's typically I think in the mid 15, high 15 kind of percent range bioavailability range. So that's why we went after berberine, because we just knew it's such an amazing ingredient, and even with its low bioavailability, it's very effective. It's incredible. But why not optimize it?
Why not optimize it? So
Yeah.
I've heard the discussion out there before, because liposomal ingredients have greater bioavailability. You could actually use less of it than its non-liposomal counterpart.
Yeah.
Your comments and thoughts on that?
I agree with that. We actually did plug that into a recent study. Like, yeah, you're supposed to use isomolar amounts, right? But we wanted to kind of do a little marketing spin on one of our studies,
Sure.
to actually compare an exact dose versus a non-liposomal higher dose. And we will be sharing that soon. It's going to be very relevant and practical for the end consumer. So you can really see what is the dose I can decrease to.
So I'm just going to ask you, when you get that data, when you get that as a study, I assume that you'll be looking to publish it. Once that happens, make sure to share it with me, because about every 2 weeks or so, I publish my Vitamin Professor newsletter, in which I summarize 3 interesting nutraceutical studies that are recent and relevant to industry, and I'd love to include that in there.
Oh yeah, absolutely. Definitely my pleasure. Would appreciate the opportunity. I mean, it's again just getting this out to the end consumer so they can see how much less of a dose they can take. And that's where you can offset some of the increase in cost as well with liposomes. Like liposomal glutathione, one of our best-selling liposomal ingredients. There's no exact dose that people take, but typically a serving size is 500 milligrams, and maybe they take it 1, 2, 3 times a day. Our customers, based on the data that we have, they'll reduce it, and this is still a nice strong dose. They'll reduce it down to 300 or 350 milligrams. So they're less glutathione, offsetting that cost, and it can be pretty damn competitive. And now you're getting a way more effective ingredient for the same or slightly more price.
You know, I have to tell you a couple of things that I think were particularly important, what you talked about today. One of them was the stability issue. You know, for those of us who have been in the industry for a long time, especially those who have worked at or very closely with contract manufacturers, we know the issues of stability. Not, stability certainly just in the product itself, but also, in the body. and to be able to have the sort of double blessing of
Yeah.
improved bioavailability and improved stability, that is huge. And then, of course, the other thing I've already commented on, but it bears repeating, is that you're able to show a connection not just with bioavailability but efficacy as well. I mean, that those are two huge things that aren't always addressed, and so again, that's pretty impressive. Now, you've mentioned a couple of the studies. I know you've done some newer studies. Can you mention what those are and what those are about?
We really, drive R&D and clinical research, preclinical research like no other company in the industry. We always have from the start. We always have a huge amount of research going on, and I would say just in the last 6 months, maybe less, maybe 4 months, we've had the second published study on our Nordic cherry, our tart cherry extract. This was a really neat and very focused study for us where it was a metabolic health study where we were looking
Okay.
at uric acid levels. We saw a statistically significant decrease,
Nice.
and it's, to my knowledge, it's the first powder tart cherry study to show this effect. People often try to make this huge jump from these liquid tart cherry
Right,
studies.
right, and somehow, yeah, it's like the ones who are doing apple cider vinegar
Right.
gummies or whatever. It's like it's not the same as drinking
Not the
apple
same.
cider vinegar. Not that there's a ton of research on drinking it, but there
Yeah.
is some. And
Yeah.
but suddenly making that translation to powdered that you put in a
Correct.
you know. So
Correct.
yes, you're
A
right.
huge difference. It can't be translated. So, really powerful study on our Nordic cherry is published, peer reviewed and published. One of the studies I was really excited for was a joint support study on our ginger
Yeah.
T3. Yeah, it's a super specialized, highly, by far the most potent ginger extract in the industry. And it's not just a high gingerol percentage. It's a very specific composition where we dialed into certain gingerols, shogaols, and some other compounds, all to target these three areas in the body, which is why we call it ginger T3. So it has the anti-inflammatory effects. It actually targets an analgesic target, vanilloid receptor, and we
Really?
were looking to see, yeah, can we heighten these compounds, get this very quick, how fast of an effect could we get from it, and how potent of an effect could we get? So you have everything from like WOMAC scores in there, and we were seeing stuff within 48 hours, sometimes less statistical significant differences. So really meaningful, really fast. Anybody that wants, looking for joint support, and it's a super small dose. It was just 125 milligrams, so really potent
That's
stuff.
tremendous. Just I don't know if you remember off the top of your head, but do you remember any of the inflammatory markers that you saw affected?
We looked at, and there were a lot in there, but there was your typical like CRP. There was, God, I should remember some of the other
No, that's
ones.
okay. I mean, you
Yeah,
do so many studies, I don't expect you to remember all the details off the top of your head. I was just particularly interested. I was at a, uh, a longevity biohacking conference in China, in Shanghai,
yeah,
late last year. And what of all the different professors and all the people presenting, one of the recurring themes was that the role that inflammation plays in
yeah.
aging and disease kept coming over and over again, and they talked
Oh
about a
yeah.
number of different inflammatory markers. So that's why I was just wondering about that. But I got is that one been published as well?
It is published, and I just pulled it up because I get rusty on some of these.
You got to send me a copy of that. I want to
I
take a look at
will.
that.
We got IL-6, TNF
Okay,
alpha,
yeah,
C-reactive protein. It's a whole like nice gamut
beautiful.
of them. Yeah. So and exactly, all these aging, disease progress. There's inflammation is always at the heart of all
It
of them.
is.
So
It is that.
addressing inflammation is a big thing for us. But yeah, super pumped about that joint support study. and how small of a dose it is, and how easy it is to formulate with. So that was another one.
You've given me some ideas because, of course, my primary role professionally is as the chief scientific officer of Nutraland USA, but I do some work in formulating products out there for some different brands, and I'm always looking at what are some of the newer, sexier things, and I'm getting a lot more people who care deeply about coming out with real stuff, not fairy dust, but real nutraceuticals. And in our conversation, you've given me a few ideas, so we have to talk.
Yeah, it's probably share a lot of the same passions. We just want ingredients to work. We want people to take them and not wonder, did this work? Did this help me or not? Like for me, that's the thing I hated the most. Back in my early 20s, I worked at my grandmother's health food store and sold a lot of the brands and the products that were out there. Yeah. And I would always, ask the customers when they'd come back, how did this work for you? How did this? I would quickly learn there were some ingredients or some brands that people would be like, this was, incredible. It helped me this, it did this. And then other times there would be ingredients across all brands that would just be like, I don't know if it really helped. Maybe it, and that always just like triggered me. I was like, that is an amazing compound. It just has bioavailability issues, like get it into the body and it'll work. So that's always just been the focus, make them perform better and get the consumer, being blown away by the effects of these extremely effective natural product ingredients. So
absolutely. Well, let me wrap up with this question. I've asked you a number of questions and you've eloquently answered them all. Are there any questions that I didn't ask you that you think are important to address that you'd like to share with our listeners, or do you feel like you pretty much covered it?
There are probably two questions that I would raise. So, and it kind of builds off what you had said earlier. You're obviously very aware of the stability issues of ingredients, both in the product itself and as it goes through the body before it even gets into the blood. I would say think about like the different formats and how much they can affect stability. And this is a huge issue in our industry, and this is something we're working on. Liquid formulas are a disaster in this industry, with few exceptions.
Right.
Because the companies are not formulating the liquid formulation to keep the active ingredient stable over time. It's something we've been working on with different customers, because they would initially ask, can liposomes be stable in these liquid formats? And full disclosure, if it's a water-based formula, it's the most difficult format to keep liposomes stable, and a lot of the times it's not possible. If the water concentration is too high, it's not possible. They will just disintegrate over just literally days. But we have been working on and developing from the ground up liquid formulations to keep liposomes intact, as well as helping that ingredient be more stable as well. So think about when people take these liquid products, like be very aware that it is a much more challenging format than a dry format, or even like a glycerin-based like emulsion. Those can keep things especially liposomes, they can keep liposomes much more stable. So be very aware of liquid formulations out there. A lot of ingredients can never be stable in them, like creatine. It cannot sit in water. We've known this for 30 years, and they constantly try to attack it. So be aware of that, especially when you see a water-based liquid liposomal formula. The chances are highly unlikely. I'm not going to say impossible, but highly unlikely there are any liposomes in those formulas. We've been testing them for years. I'll just leave it at that. So
a very, very good point. Now, you said you had a couple of questions.
Yeah, one, and this one's just kind of more like entertaining, funny. But I used to get this question asked early days when I was doing liposomal talks, which is, how do you get the ingredients to be nano size? Because your liposomes are typically nano size. And people have this idea that a vitamin, like take vitamin C, for example, is so massive and somehow we're going to shrink it down and make it nanosized into a liposome. So I just started reeling off these details early on because I realized the industry as a whole didn't properly understand this. So take vitamin C, for example. Vitamin C is 598, 600 angstrom or basically 59 nanometers. So it's already way smaller than our 100 to 200 nanometer range on the liposome. So they're naturally nano size for the most part. It's not that we have to destroy anything or chop anything down to make it nano size to fit inside a nano liposome. And then the second piece to that is just because a single liposome and the contents of it is nano size, that does not mean the powder, the mesh size of your finished product is nano size.
Right.
It's not even close. So understand one little particle of your powder has hundreds to thousands of these nano-sized liposomes in there. So I just wanted to kind of mention that too.
Well, those are excellent points. Thank you for sharing that. And Sebastian, thanks so much for joining me today. I feel like I got a whole new education on liposomes. I've certainly learned a few new things about your particular LipoAdvantage or LipoVantage delivery system. And I just wanted to, again, thank our listeners for joining us, and to ask you again, if you like what you heard and saw, don't forget to like and subscribe. So thanks again, Sebastian. I'll hopefully see you at Expo West coming up soon.
Thank you, Gene. Appreciate it.